Good morning, everyone, and thank you very much for coming. First, I want to thank the organizers for inviting us to present, and I also want to thank Katie for introducing this topic, as I'll be touching upon many of similar market opportunities here. So Cellerant Therapeutics is a company located in San Carlos, California, in the San Francisco Bay Area. Our focus is to develop immunotherapies, novel immunotherapies for blood-related cancers and blood disorders. We have a couple of programs. The first program is romyelocel-L. It's a universal, off-the-shelf cellular therapeutic that's designed to provide innate immunity during neutropenia. We've completed a randomized, controlled phase 2 trial in AML patients, and we've demonstrated efficacy in that trial. We completed our end of phase 2 meeting with the FDA, and we have a phase 3 trial design agreed upon with the FDA. We were also granted the RMAT designation for use of this product in de novo AML patients by the FDA about a few months back. And as you heard from a previous speaker, the market opportunity is significant. It's beyond $1 billion in AML, hematologic malignancies, and other solid tumors. The second program is an antibody-drug conjugate program focused on leukemic cancer stem cells and potentially to cure AML. The target here is CLL1, also called CLEC12A, and it has a much better safety profile than CD33 and CD123. The filing of INDs targeted for first quarter of 2019, and then we'll initiate a phase 1-2 trial soon after. I will not be talking much about that program besides this slide. We have been very efficient in raising capital over the past 10 years. We've raised $25 million in equity and convertible notes, and we've received over $170 million in non-dilutive funding from the US government. So this is a pipeline of our product. Romyelocel-L is ready for phase 3. We are preparing to initiate our phase 3 trial. We are working with BARDA to develop acute radiation syndrome for the application of acute radiation syndrome, which is approved under the FDA's animal rule. We have another indication for romyelocel-L. It is to induce tolerance in organ transplants, and we've done some preclinical studies to demonstrate that it can induce tolerance in certain settings. And the reason for that is, obviously, these cells have the potential to make some of the regulatory cells, such as myeloid suppressive cells, as well as dendritic cells. And then, as I mentioned to you, the antibody-drug conjugate program will be in phase 1 early next year, and we have another antibody-drug conjugate target, IL-1RAP, that's in preclinical stage at this point. So with that, I'm going to move on to talk about romyelocel-L and describe the product, our phase 2 results, and then talk about our phase 3 design. So it's a universal off-the-shelf cell therapy product that is designed to provide innate immunity, thus to prevent bacterial and fungal infections in patients with prolonged neutropenia. Neutropenia, as you know, is a significant issue in many of these patients who receive myelosuppressive chemotherapy. This myelosuppression induces significant level of reduction of myeloid cells, and that contributes to, obviously, high risk of infection. And because of that, suboptimal treatment is normally given to these patients, and their chemotherapy is not continued. So it's a significant unmet medical need. Yes, G-CSF is approved for this indication. However, it's not commonly used because G-CSF requires these progenitor cells to be present for them to actually bind to and make neutrophils. In the absence of these cells, G-CSF is ineffective. So as you can imagine, it adds substantially to the cost of care, hospitalization, and antimicrobial therapy. The market opportunity is significant. It's over $1 billion. Just AML alone, the incidence is 20,000 patients, 50,000 prevalence. And all AML patients, whether they are in induction, consolidation, or they're receiving older patients receiving hypomethylating agents, they all have prolonged neutropenia. And in stem cell transplants, like cord blood transplants, the ANC recovery neutrophil recovery is significantly longer as well. And other hematologic malignancies and some solid tumor patients who receive myelosuppressive therapy also have prolonged neutropenia. So this particular product has indication far beyond AML. So what's the product? The product is primarily a CD34-derived, mobilized peripheral blood CD34-derived hematopoietic progenitor cells. These cells are of myeloid lineage. They do not have they have minimal or undetectable T cells. And the process of production is very simple and very robust and economical. We have demonstrated reproducibility from between donors and between lots, and we've produced now over 25 to 30 GMP lots at recognized contract manufacturers. We produce quantities such that a large quantity can be sufficient for at least a part of the clinical trial. And we have 3 years of shelf life data, at least so far, from GMP product that we've used in our phase 2 trial. The product is very patient and provider friendly. There is no HLA matching required. The product is available as and when required. It is delivered cryopreserved to the hospital bedside, thawed by the bedside, and infused by simple IV infusion. We've demonstrated safety of this product in over 150 patients in 3 different clinical trials. So this is the design of the randomized phase 2 trial that we completed. It's an open-label, controlled, multi-center randomized trial. 21 sites in US participated in this trial. I'm going to share with you data on 78 de novo AML patients who received induction 7+3 chemotherapy, which is a standard induction therapy given to these patients. The treatment group received our product on day 9, 10, or 11. That is 2 days after the last dose of chemotherapy. Chemotherapy goes for 7 days. And they received GCSF on day 14. The control group also received GCSF on day 14. There were no restrictions on use of any antibiotics, prophylactic, empirical, or treatment on both the groups. And the reason why GCSF is given on day 14 is physicians don't like to give GCSF before they actually do the bone marrow biopsy of these patients. Infections were adjudicated by an independent blinded experts. They did not know whether it was from control or treatment group. Every infection was adjudicated by them. Patients were followed for 42 days, and endpoints I'm going to talk about today include infections, hospital days, and antimicrobial use. And based on our discussion with key opinion leaders in the field, it's clear that anything over 20% reduction in infections would be very significant and meaningful for these patients. So before I talk about the results, let me briefly take you through the mechanism of action. So these cells, home to the bone marrow, differentiate and migrate to tissues back. They go through the blood, go back to tissues. That's where neutrophils normally reside. And romyelocel is infused on day 9, and we know this based on our cord blood data that I show in the graph at the bottom here. So it takes about 4 or 5 days for the cells to come through circulation, come back into the tissues. This was done through chimerism in our phase 1 cord blood study. And GCSF is given on day 14. So by the time they're ready to come out into circulation, with GCSF, there will be a boost of neutrophils. So we expect full biological activity to be observed from day 15 on. And incidentally, the high risk of infection in these patients is also around that time. These patients have their marrow almost depleted by day 14. So they have prolonged neutropenia because of that reason. And as the IDSA guidelines point out clearly, that if the risk of infections are much higher when you go beyond 7 days of neutropenia, which would imply that you don't have the progenitor cells to make neutrophils. And so during that period of 7 days, they've also received prophylactic antibiotics or empirical antibiotics or antifungal agents to treat those infections. And organisms that survive during this 7 days period and come back are usually more drug resistant and mutant organisms and difficult to treat. The only way to clear those infections may be innate immunity, maybe through innate immunity of the system, of our own body. Thus, day 15 to day 28 is a critical period for these patients because that's when they struggle the most. So these are the results from our phase 2 study. This slide shows decrease in infections. I'm showing you 2 different time periods here. Day of infusion through day 28 and from day 15 to day 28. There was a 35% reduction in serious infections, MDI or CDI, during the period of day infusion to day 28. And if you look at day 15 to day 28, we saw 73% fewer patients had serious infection. A significant reduction. The p-value is significant. And in the interest of time, I'm not going to the details of the antimicrobial data, but we saw 44% fewer patients receiving antimicrobial therapy to treat infection. Similarly, the intensification of antimicrobials due to neutropenia, due to febrile neutropenia, was also decreased around 45% in these patients compared to the control, sorry. So we saw a 3 days decrease in hospital with a p-value of 0.006. And this decrease is primarily due to the number of infections in these patients, even after they recover neutrophil. So if you look at these 2 notes at the bottom, 10 patients in the control group had infections beyond day 21. And this is when they actually recovered the neutrophils. So the infections they have probably were residual infections from before that didn't get cleared, that became more pronounced after day 21. In the treatment group, we did not have any patient that had infections beyond day 21. So that would explain why the patients just stay longer in the hospital during that period of time. So we did not see any imbalances in SAE, safety serious adverse events, between the treatment and control groups. We had a data safety monitoring board review this trial continuously, and there were no safety concerns. The couple of points I want to make here. Of the SAEs we saw 8 in the control group and 9 in the treatment group. 7 out of the 8 in the control group were infection-related. 4 out of 9 in the treatment group were infection-related. We saw a total of 3 deaths in the study. 2 deaths were due to infection in the control group. There was 1 death in the treatment group due to progressive disease. So clearly, we are seeing the same indication with the SAE safety profile as well in these patients. So we presented the phase 2 data at ASCO this year, and then we had an oral presentation at EHA this year where we talked about our results from 7+3. What I'm not talking about today here is that we had another cohort in this HIDAC, which we plan to present. That data we presented at ASH this year. So after we finished our phase 2 studies, we met with the FDA. We agreed on a plan for a phase 3 design. So there'll be a single phase 3 study, pivotal phase 3 study for this product, and it will be a similar patient population, 7+3 de novo patients, 55 years and older, and receiving standard chemotherapy. Okay, the primary and secondary endpoints would include serious infections and days in hospital. And all of the infections will be adjudicated by an independent committee. In fact, this time we plan to even have the antimicrobial analysis done by this independent committee. So our intellectual property is quite vast. We have a portfolio of patents that are either owned by us or exclusively licensed from Stanford University from the labs of Dr. Irv Weissman and Dr. Janice Brown. And these include use of allogeneic myeloid progenitor cells without HLA matching to provide myeloid function, use of these cells derived from multiple unmatched donors to improve impaired hematopoiesis, and method of making therapeutic composition for transient hematopoiesis, and also to make from multiple stem cell sources, and a method for enhancing engraftment of allogeneic hematopoietic stem cell graft. I didn't describe a lot about manufacturing. The manufacturing is very straightforward. We collect CD34 cells. We actually collect mobilized peripheral blood. We isolate the CD34 cells. We bank the CD34 cells from a number of different donors. Day of manufacture, we pool multiple donors, expand them, and then we cryopreserve the product. So it's a very simple, straightforward expansion process. And as I said, we've done this so many times now. It's very robust. The process is extremely robust. So in conclusion, hope I've conveyed to you that we are developing some immunotherapies that are significant, that cater to some unmet medical need. The off-the-shelf cellular therapy designed to provide innate immunity has demonstrated efficacy in phase 2, and we have a clear path forward, and we've been granted RMAT designation. The market opportunity is significant, and the antibody-drug conjugate, if anybody's interested, we are willing to talk. We're happy to talk about it. Again, it's a very, very potent target compared to CD33 and CD123. And as I mentioned to you, we have been highly efficient in managing our capital, and we're in the process of raising capital and looking for partnerships ex-US at this point. Thank you very much. I'll be happy to answer any questions you may have.